everyone. Welcome to a new session of the paper Molecular Biology. Here we discuss DNA recombination, replica plating and Ames test. The integrity and stability of DNA is essential for the sustenance of life. Recombination is a type of DNA rearrangement which alters the combination of genes present in any individual genome as well as the timing and the level of expression of these genes. Exchange of genetic material in DNA recombination can happen between multiple chromosomes or between different regions of the same chromosome. The recombination takes place between homologous regions of chromosomes. Genetic exchange needs some degree of sequence identity. In diploid eukaryotic organisms, the exchange of genetic information takes place during the process of meiosis. Here, the genetic recombination is to ensure that each gamete includes both maternally and paternally derived genetic information such that the resulting offspring will inherit genes from all four of its grandparents, thereby acquiring a maximum amount of genetic diversity. In a population, this type of genetic variation is crucial to allow organisms to evolve in response to a changing environment. The DNA rearrangements are caused by a set of mechanisms that are collectively called genetic recombination. Recombination lends the genome flexibility. Without genetic recombination, the genes on each individual chromosome would remain fixed in their particular position. Changes could occur by mutation only, which would be hazardous. Recombination provides the means to achieve extensive restructuring, eliminate unfavorable mutation, maintain and spread favorable mutations, and endow each individual with a unique set of genetic information. This greatly enhances the evolutionary potential of the genome. Recombination must occur between precisely corresponding sequences in the case of homologous recombination to ensure that not one base pair is lost or added. The newly combined or recombined stretches of DNA must retain their original structure in order to function properly. Two types of recombination can be distinguished. The first one is generalized or homologous recombination, and the second one is site specific recombination. In generalized or homologous recombination occurs in eukaryotes during meiosis. A third process, transposition, is a type of recombination in which DNA sequence is inserted into another place without regard to sequence homology. Other applications of recombination include DNA repair, as in the case of double-stranded breaks, filling gaps during DNA replication for preventing stalling of the replication fork. Here, normally sister chromatid serves as the donor of missing material through recombination followed by DNA synthesis. Bacterial recombination was experimentally proved by the microbiologist Joshua Lederberg in the bacteria E. coli in 1947. Barbara McClintock and Harriet Creighton in 1931 through experiments with maize demonstrated the role of recombination during the inheritance of chromosomes. Two general models can be distinguished to explain homologous recombination: recombination initiated from a single strand DNA break and recombination initiated from a double stranded break.